So here is another cute story about nature's rules and biological loopholes. Obviously, global warming is a big concern, especially ocean warming and heat waves for corals, coral bleaching, and so on. And yet, we have to look for signs of species that may be better at responding to the warming or even surviving and thriving in warming waters. So this paper that just came out looks at genetic techniques to figure out how the role of gene expression and symbiosis in reef-building coral acquired heat tolerance happens. So they find 11 different locations of Great Barrier Reef. They take the samples and look for genetic response and response to symbiotic algae that live with them to see where the heat tolerance comes from. So basically they crossbreed different species with different symbionts and look at the heat tolerance response in terms of genetics and symbiosis at the larval stage, which is more dominated by the genetic expression to heat stress, versus juvenile stage where the response is now affected by symbionts and so on, and conclude a few really interesting things which could help the future protection efforts, coral gardening, to make some of the corals better equipped to survive. Predicting how reef-building corals will respond to accelerating ocean warming caused by climate change requires knowledge of how acclimation and symbiosis modulate heat tolerance in coral early life history stages. So they may have a genetic ability to respond to ocean warming, and then they, if they survive past the larval stage into recruitment, become adults. Then they also respond to symbionts who themselves may be more heat tolerant or less heat tolerant species of phytoplankton. We assayed transcriptional responses to heat in larvae and juveniles of 11 reproductive crosses of Acropora tenuis colonies along the Great Barrier Reef. So they take 11 samples and then cross them to look for intrinsic genetic response versus response to symbiosis. Very nice work. Even though I'm not an expert in ecology, I'm just conveying the basic message from the paper. Larvae produced from the warmest reef had the highest heat tolerance, so they have evolved over time. Although gene expression responses to heat were largely conserved by cross identity. So even though intrinsically the ones in warmest waters were responding genetically to heat warming waters or perturbations of heat, the others were also able to respond, and the response was possible even in the corals that were not living in the warmest waters. Juvenile transcriptional responses were driven strongly by symbiosis. When in symbiosis with heat evolved Symbiodiniaceae, hosts displayed intermediate expression between its progenitor Cladocopium and the more stress-tolerant Durustinium. So the algae, zooxanthellae as they are generally referred to, that live on shelled organisms like corals, can be less heat tolerant like the Cladocopium or more stress tolerant like Durustinium. So the larval response is transcriptional genetic related, and then juveniles have already the symbiotic impacts, indicating that the acquisition of tolerance is a conserved evolutionary process in symbionts. Okay, heat evolved Symbiodiniaceae, I have trouble saying this word, facilitated juvenile survival under heat stress, although host transcriptional responses to heat were positively correlated among those hosting different genera of Symbiodiniaceae. These findings reveal the relative contribution of parental environmental history of the corals themselves, as well as symbiosis establishment and coral molecular responses to heat in early life history stages. So obviously the molecular level response in the larvae and the juveniles are different, but the symbionts themselves have also adapted to how the corals respond to heat, even though this is not fixed, in the sense if you crossbreed, those from less tolerant regions are able to respond to stressors with the genetic and symbiosis response, which obviously can be used to make plans for future to reduce the vulnerability of corals which are not doing well. Okay, so this is showing a general picture of how the corals look like. This is from an Economist article which looked nice, so I took it. Obviously lots of colors. These are soft corals waving in the water, but there are also hard corals which have structures that we'll look at in the next slide. But corals which occur typically in warm waters, there are also cold water species which are not reef building, but let's focus on warm waters where the warming is maximum under anthropogenic forcing. These are affected by acidification and warming, and the warm waters with less nutrients, corals produce a structure which hosts photosynthesis, which gives them these colors, and then the photosynthesis produces oxygen, takes CO2 produced by the corals, and then the nutrient cycling, etc., is supported by the habitats created by this ecosystem for very divergent species of small and big fishes. So this is like the rainforest of the ocean, and they do occur in warm waters, which are generally nutrient poor, but they have this symbiotic system which makes it possible. Corals are, of course, little animals. Coral polyps have these kind of structures with tentacles and stomach and mouth and outer epidermis, septum, and so on. So when global warming with warming temperatures and acidification begin to harm the corals and bleaching happens, then the substrate needed to grow new corals also gets damaged. So all kinds of feedbacks happen to accelerate the coral bleaching and the general lack of recovery of corals from perturbations. For example, during La Nina, when the West Pacific warms, you can get natural coral bleaching, or during El Niños also sometimes coral bleaching happens because of the warming. Depends on where you are and so on. But the recovery from such natural perturbations is going to be affected by global warming and acidification, of course. So here are the warmest temperatures, the Indo-Pacific warm pool. Here is where you get the so-called coral triangle, but you also get corals here in the Gulf of Mexico, which is very warm, as well as the Red Sea and the Persian Gulf and so on, as you can see here. So you can see the warm water corals all over these regions in the Indian islands here, Andaman, Nicobar, Lakshadweep, Sri Lanka, down to Maldives, and around Madagascar and into the Gulf of Mexico. And of course, the coral triangle here into South China Sea, up into the northern parts of the South China Sea, East Sea, whatever it's called by each country, and the maritime continent, Australia, and the Western Pacific warm pool. Of course, waters get quite cold here, but still you have some corals in here around Galapagos, of course. NOAA has these coral bleach warning systems which work like this. So this is a map of tropical coral reef locations, of course, and this is the numbers of observed bleaching events identified by Coral Reef Watch program by NOAA, with a 50-kilometer satellite monitoring during the comparison time period of August 1, 2011 to December 27, 2015. That ran right through the biggest El Nino of this century, 2015-2016. So you can see that the number of events, number of observed bleaching events has been high in many places and lower in other places. So you have all kinds of ocean dynamics happening to determine in the net where coral bleaching happens. So it's a brief story. I'm not going to show any of the results because they are kind of complicated analysis of transcriptional response in the larvae and transcriptional and symbiosis responses in the juveniles to see how the overall heat tolerance occurs and whether it is just related to the genetic intrinsic genetic ability to tolerate heat, or whether there is possibilities of doing genetic engineering to provide better heat tolerance. Okay, so the discussion goes as this: heat tolerance may be regulated via several divergent evolutionary pathways, including genetic fixed effects or via changes in gene expressions. So genetic ability may exist, and environmental perturbations may make them express themselves like the phenotypic response during the symbiosis interactions. These mechanisms are likely linked, where the interplay between the genetic fixed effects and gene expression can be broadly connected by mechanisms like expression QTLs, which is quantitative trait loci, how parts of the genes express the traits. Here we use a unique experimental design to quantify the synergistic effects of acquired heat tolerance via gene expression plasticity and symbiosis in early life history stages, so larval versus juvenile responses. We profiled gene expression in both aposymbiotic, which means when the symbionts and the host are living separately, well, both are symbionts in that case, aposymbiotic larval and symbiotic juvenile stages at pre-stress, ambient and heat stress treatments across 11 distinct genetic crosses between and amongst northern and central Great Barrier Reef sites of Acropora tenuis. We found that the selective breeding between disparate sites revealed relatively conserved gene expression profiles, underlying key differences in larval and juvenile heat tolerance between crosses. So they took different locations and crossed them to see how much the genetic fixed trait versus genetic expression matter to environmental perturbations. So underlying key differences in larval and juvenile heat tolerance between crosses, although mechanisms of heat tolerance differed substantially between these disparate life history stages. So larval stage and juvenile stage molecular response mechanisms are different. Juveniles hosting SS1, which is one location under ambient conditions, exhibited an intermediate heat response situated between D1A, which is a Durusdenium, and their progenitor taxa C1, which is the Clepodenium, or whatever the name I forgot. Clepocodipian, whatever. Sorry. These results suggest that the evolution of heat tolerance in the SS1 strain likely follows a conserved or constrained evolutionary pattern. So they may all carrying the genetic ability to do it, but whether that gets expressed or not depends on where they are living and who the symbiont is. In Symbiodiniaceae, given the recapitulation of molecular host responses to SS1 and D1A, but residual or ancestral similarity of SS1 to C1, it is likely that further experimental heat selection may be needed to completely convert the SS1 capacity for heat tolerance that is in line with the canonically stress-tolerant Durusdenium species. Together, these data quantify and simultaneously the relative contribution of gene expression and symbiosis in critical early life stages like larval to juvenile transitions, and provide an applied foundation for optimizing genetic-based methods for enhancement of coral survival. So genetic engineering is being talked about here. Nonetheless, the name, let's get it right, is Cladocopium and Durusdenium. Okay, this is more stress tolerant, this is less stress tolerant. So where they live and how they have co-evolved with the coral host determines their stress tolerance, but it is easy to crossbreed them and provide enhanced stress tolerance as seen in these lab experiments. Okay, very, very nice work. So I just wanted to include it here, and let's leave it here. Remember, we have talked about stress tolerant corals elsewhere as well, but generally I'm very keen about how ecology manages to survive global warming, ocean heating, ocean heat waves, and refugia that are created in certain cases where heating, like the Galapagos penguins that are surviving and maybe even expanding in population because of some refugia that is created in the larger scale warming with local upwelling that creates cold pockets of water and so on. I have reported on that Galapagos penguins elsewhere. Okay.